Hi, today we're going to be talking about neurons, impulses, and the synapse. Our guiding questions for today are: what is the structure of a neuron, and how does this relate to its function? How is the resting membrane potential established by ion pumps and channels? How is an action potential generated and conducted across a neuron? And how does the synapse mediate communication between neurons? So today is going to be a zoom in into the level of a neuron and how that works on a cellular level. And next session will be a zoom out into the nervous system as a whole and how the parts interact with each other. So our nervous system is split up into the central and the peripheral nervous system. Central nervous system consists of the brain and the spinal cord. Some people think we only use 10% of our brains, but that's obviously not true. It doesn't make any sense from an evolutionary perspective, so anyone that thinks that probably needs to use more of their brain. The peripheral nervous system consists of all the peripheral nerves that are delivering information to all the parts of your body, your muscles, the receptors on your skin that you use to sense touch on your skin, and all the other sensations. In general, we know that the neurons in the peripheral nervous system can do some degree of spontaneous regeneration after they're cut. But in the central nervous system, very little of that happens. So that's why traumatic brain injuries and spinal cord injuries are so serious. A neuron is an individual unit of the nervous system that carries information throughout the body. A bunch of neurons wrapped up together with a blood supply and a sheath to hold it all together, that is what we'll call a nerve. So our body uses both sensory input and motor output to coordinate its efforts. Our nervous system works by forming paths between the brain and the body. So we'll have some sensory information from the eye as we see this glass of water, and that information will be passed by an electrical signal through neurons that lead into the brain. And in the brain, this information is integrated together with other information the brain is receiving. This information passes through many distinct centers of the brain for processing, and then finally the brain decides on an action. It initiates this action by sending a signal through the neurons that lead to the muscles of the arm in this case, and that signal will be to tell this muscle to contract so that we can bring the glass of water to our mouth and drink. So it's a relay system. When we look at the structure of a single neuron, so this is a single cell, we have three parts: the cell body with a nucleus, the dendrites, and the axon. It's the job of the axon to send a signal, and the axon can be up to 1 meter or 3 feet in length depending on the particular neuron. So it's quite a feat for it to be able to conduct a signal this far on a single cell. The axon is acting like a wire, just like an electrical wire. The signal is going to be in the form of an electrical pulse, a momentary pulse that's going to travel all the way down this axon. And what's it going to do when it reaches the end of the axon? It's going to transfer that signal to the next neuron in the chain. The end of the axon meets the dendrite of the second cell, and this area where the two meet is called the synapse. Although the signal is being transmitted by electrical pulses all the way down the axon, the electricity is not able to cross this little gap called the synaptic cleft. So when the signal reaches the end of the axon, the synaptic terminal, it changes from an electrical signal into a chemical signal. That electricity is going to release the neurotransmitters, which are small chemicals stored inside vesicles. They're stored ready to go, and when they receive that electrical pulse, they release the chemical into the cleft. This is a very small space, so the chemicals very quickly diffuse to the other side, where they're going to elicit a response in the second cell. This is called the presynaptic cell and the postsynaptic cell. And what the postsynaptic cell is going to do is these chemical signals are going to tell it to initiate a electrical pulse of its own, and thus that electrical pulse will pass down the axon of this cell, moving on in the chain to the next cell until the signal reaches its destination, which could be either the brain or perhaps a muscle cell. So axons send the signal. Dendrites are listening to the signals being received by the axons. Notice that this axon in the picture is covered in a myelin sheath. Myelin is a fatty substance that wraps around the axon to insulate the electrical signal. It's the same way that electrical wires can be insulated by rubber. Each of these little pieces of myelin is actually an entire cell. These are called glial cells, and the job of the glial cell is to wrap around the axon to produce lots of fatty myelin to insulate it. Only some neurons are myelinated, not all of them, but the ones that are myelinated have an increased conduction speed. Having this insulation prevents electrical charge from leaking out of the axon. Another thing that changes the speed of the axon transmission is its diameter. So a larger diameter axon will transmit its signal more quickly. If we're looking at speeds of these things, the smallest unmyelinated neurons will have a conduction speed of 0.5 meters per second. All the way up to the largest myelinated axons, they will conduct at 150 meters per second. And in case you want to know about the size of the axons as a whole, in our bodies they are from 4 to 100 microns, and so that's 4 1,000ths of a millimeter up to 1/10 of a millimeter. In the giant squid, however, these axons can be up to 1 millimeter in diameter, so about the thickness of a very thick strand of hair. The giant squid axon was gold for researchers, because finally they had a neuron that they could isolate, that they could see, and that they could stick electrodes in to measure how it transmits its signals. In the nervous system, in addition to the neurons, there are glial cells, and the glial cells provide various kinds of support, either creating myelin like this oligodendrocyte is doing, or other support functions. Some provide physical support to the neuron structure, some provide maintenance and repair for those cells. So know that they're there and supporting the neurons. If we want to talk about how the neuron sends its electrical signal, we first need to look at what the neuron is like when it's at rest. Here's a resting neuron, and we're going to be looking at the relative charges inside and outside of the membrane. There is a sodium-potassium pump, and for every cycle of this pump, it's going to be transporting 3 sodium ions out of the cell and 2 potassium ions into the cell. That's what it does on every turn of its action. Naturally, the concentrations of sodium are higher outside of the cell, and potassium is higher inside the cell. So this pump needs to be working by active transport. It's transporting these ions against their concentration gradients, and the molecule it uses to get that energy is ATP. Now there's an easy way to remember these concentration gradients. K plus potassium has 2 characters, same as In for in, so there's more K plus on the inside of the cell and more Na plus on the outside. Notice that this sodium-potassium pump, so it's pumping 3 positive charges out for every 2 positive charges in. So on average, it's pumping positive charges out. This pumping action contributes to the inside of the membrane being negative compared to the outside. This is what's called a negative membrane potential, and the resting membrane potential is always negative. If you forget what the sodium-potassium pump does, you can also use this to remember. The pump moves 2 K+ into the cell for every 3 Na+ out of the cell. Since it's generating an electrical potential difference, we call this pump electrogenic. As it keeps working, it's going to make the inside of the membrane more negative and the outside more positive. In addition to this pump, we have a different channel called a potassium leak channel. Since there's more potassium on the inside of the cell, this channel is working to allow potassium ions to leak out, so it's also contributing to the inside's loss of positive charges. It's also contributing to the net negative charge inside the membrane compared to outside. This leak channel, however, is passive transport. It doesn't require ATP. Both of these pumps are contributing to a negative resting membrane potential. And know that there are other channels called voltage-gated channels here, but they are inactive. We're going to talk more about them later. All in all, we have the maintenance of a negative charge on the inside of the membrane compared to the outside. We call that the resting membrane potential. And if we put in 2 electrodes and measure, the potential difference will be about -70 millivolts in most neurons. Millivolts is a measure of the potential difference, and this V subscript M, that means the M is for membrane, so this is the membrane potential, V for voltage. Now it's important that you know that I'm not talking about the charge of the entire cell. I'm talking just about the charge directly inside the membrane, out here, and directly outside the membrane, out here. That is what we're measuring. If we measured the charge of the cell as a whole, it might not be net negative. But this area directly beneath the membrane compared to directly outside the membrane, there is a potential difference of -70 millivolts. Okay, we're going to watch an animation to review the concept of the resting membrane potential.
A nerve impulse is an electrical current that travels along dendrites or axons due to ions moving through voltage-gated channels in the neuron's plasma membrane. Voltage-gated channels open and close in response to an electrical voltage, so they are affected by changes in electrical charge around them. When a neuron is at rest, a charge difference is maintained between the inside and outside of the cell. This charge difference is produced and maintained largely by active transport using sodium-potassium pumps. The pumps send sodium ions out of the cell and bring potassium ions in. While other channels allow some flow of potassium ions back out of the cell, the sodium ions cannot easily get back in to replace the lost positive charges. The overall result is that the exterior of the cell has a net positive charge and the interior has a net negative charge. The difference in charge between the interior and exterior of the cell is called the resting membrane potential.
Now that we've covered the resting potential, we can go on to the actual electrical pulse that conducts signals down this axon. That is called the action potential. An action potential is generated when a cell that is at rest depolarizes, so its membrane potential goes up closer to 0 and even overshoots 0 up to about +30 millivolts. This is the height of the action potential. The membrane will then repolarize back to resting potential. This all happens on the order of several milliseconds. Now remember that this membrane potential is talking about the inside relative to the outside. So if at rest the cell is more negative on the inside, at the height of the action potential it's actually slightly more positive on the inside than the outside. We're going to do this one step at a time. Right before the action potential starts, the cell is at its resting membrane potential, -70 millivolts, and we have this sodium-potassium pump that's always working. These 2 channels are new, and these are voltage-gated channels. these are not the same leak channels that we were talking about before. They're voltage gated, and what I mean by that is that they are only going to open when the membrane potential reaches a certain threshold value. They're both currently closed because the membrane potential is too low. Now, they each have different thresholds. For the potassium channel, its threshold is negative 55 millivolts. So if there were to be some kind of stimulation that brings the membrane potential up to negative 55, at this point, the voltage-gated sodium channel will open. When it opens, remember there's more sodium outside, so that's going to cause sodium to rush into the cell. Positively charged sodium ions are going to make the inside of the cell more positive, so we're going to see a quick increase in the membrane potential. This is called a depolarization, up to about positive 30 millivolts. Now at this point, a couple of milliseconds have passed. The voltage-gated potassium channels are still not open, but at this point, the height of the action potential, the voltage-gated sodium channels will open. Why did they open so much later? They are slower to open, so the molecular action that is required to physically change the shape of that channel takes longer. When potassium channels open, that's going to allow all the potassium that was inside the cell to rush out. So the inside of the cell is going to become more negative, bringing down the average membrane potential. In phase 3 here, we see that even undershoots, the membrane potential goes lower than the resting potential of negative 70 millivolts due to the action of these potassium channels. Now at this point, during phase 3, voltage-gated sodium channels are now closed. They have closed because voltage-gated channels are gated, and they can only be open for a certain amount of time. That's their behavior. They open for a certain amount of time when the threshold potential is reached, and then after a while they close, and they enter a period where they cannot be opened again. This phase 3, as the membrane potential is going down back to resting or even undershooting it, that's called the repolarization. And once the voltage-gated potassium channels close, then we're back in that resting condition. The only channels that we have working are the sodium-potassium pump and, not pictured, the potassium leak channels. And that's going to gradually bring the membrane potential back up to the resting potential of negative 70. I'm going to move this. It's important that you know each phase of the action potential and what channels are involved in that. Resting potential, it's the sodium-potassium pump. That's the only one that's working. During depolarization, that's because a voltage-gated sodium channel has opened, and it has opened because the membrane potential has reached its threshold. Repolarization is when the voltage-gated potassium channels, which are slower to open, finally open, and they let potassium rush out of the cell. That brings the membrane potential back down, and even hyperpolarizes it in this undershoot. Once both of the voltage-gated channels are closed, the cell goes back to its resting potential of negative 70 millivolts, with only the sodium-potassium pump working. And then you should note that the potassium leak channels are working all through this, but their effect is not as great as these other channels because there's simply more of the other channels put together. I thought you might want to know how that voltage-gated channel works. So why does it only open when the cell is depolarized to a certain threshold value? It's because of the structure of the channel. For example, this is a sodium voltage-gated channel. And when the membrane has a negative potential, where it's more negative on the inside, there are two sections of the protein called voltage sensing alpha helices that hold the gate closed. They're attracted to this negative charge inside the membrane, and so they stay on this side and they're holding the gate closed. When the cell reaches threshold value, the inside of the membrane is going to be slightly more positive. It's become depolarized. That's going to repel the alpha helices away, and it's going to induce a conformational change in the protein that makes up this ion channel. The channel is now open, allowing sodium to move through it. Let's now watch the rest of this animation on how the action potential is conducted down the axon.
A nerve impulse begins when a stimulus disturbs the plasma membrane on a dendrite, causing sodium channels to open. Sodium ions flow into the cell, lessening the charge difference at that location. If the change is enough, it will cause nearby voltage-gated sodium channels to open. This allows so many sodium ions to flood into the cell at that location that the membrane there is depolarized, with the local region inside the cell having a net positive charge and the outside of the cell having a net negative charge. This affects neighboring voltage-gated sodium channels, which then open, moving the depolarization along the membrane. This moving depolarization is called an action potential. Changes occur behind the action potential to restore the resting membrane potential. The voltage-gated sodium channels close and voltage-gated potassium channels open. This allows a rapid flow of potassium ions out of the cell, repolarizing the membrane so that the inside is again negative and the outside positive. This is followed by use of sodium-potassium pumps to fully restore the resting membrane potential and to reestablish proper concentrations of sodium and potassium ions inside and outside the cell.
We just saw that an action potential, in order to conduct all the way down the axon, needs to elicit an action potential in the neighboring area of the membrane and set off a chain reaction. So if the signal is propagating from left to right, then this part of the axon will first have an action potential, and the depolarization of the membrane will be enough to spread to this adjacent area of the membrane, eliciting an action potential there, and moving down, and so on. Here, in the wake of the action potential, these areas of the membrane are undergoing repolarization. Their membrane potential is going back down to negative 70 millivolts due to the action of the voltage-gated potassium channels. But at the height of the action potential, it's really the voltage-gated sodium channels that are opening, allowing sodium to rush into the cell, causing that depolarization that we recognize as the spike in the action potential graph. There is a toxin called tetrodotoxin, which binds to and inhibits those voltage-gated sodium channels. It's found in a Japanese puffer fish, and this is something that can be actually prepared for eating, and if it's prepared correctly at a restaurant, then there is no tetrodotoxin and you can safely eat it. But if it's prepared incorrectly, then ingesting that toxin will cause an inhibition of your voltage-gated sodium channels. There will be no action potential even when the membrane is depolarized to the threshold potential. And suppose this is an axon that's leading to stimulating a muscle. Those muscles will be paralyzed. It's the paralysis of the diaphragm, the muscle involved in breathing, that causes people who ingest tetrodotoxin to stop breathing and die. If we look at the conduction of an action potential, its goal is to get that electrical signal all the way down to the synapse, the junction between the two neurons. And at the synapse, the signal changes from an electrical signal to a chemical signal. So we have the action potential traveling down the axon, it gets to the presynaptic terminal, and the way it changes into an electrical signal is by opening up these voltage-gated calcium channels. That depolarization opens these channels. They're voltage-gated, and they're found only in the presynaptic membrane right here, not along the entire axon. When they open, they're going to allow calcium, which is higher on the outside of the cell, to rush into the cell. Calcium is an intracellular signal for exocytosis. So these vesicles that are all lined up here, ready to secrete the neurotransmitter, that calcium signal is going to cause them to undergo exocytosis and release the transmitter into the synaptic cleft. This is a very small space, so these chemicals will diffuse very quickly across. They're going to reach the other side, the postsynaptic membrane, where there are ligand-gated ion channels. A ligand-gated ion channel is a channel whose opening or closing depends on the binding of the specific ligand. In this case, the ligand needs to be the chemical that is being released by the presynaptic cell. So these ion channels are going to be closed all the time until the ligand binds. When they receive this neurotransmitter, the channel will undergo a conformational change, and it will open, allowing ions to cross. In this case, we have a ligand-gated channel that allows both potassium and sodium to move in and out of the cell, just according to their concentration gradients. So this channel is going to be always closed until the ligand binds, and when it does, the channel will open, allowing ions to move across. Although both sodium and potassium are moving, the net change is going to be more sodium coming into the cell. So that's going to cause the inside of the membrane to be more positive, a depolarization. And if this depolarizes the cell enough, if it gets to that negative 55 millivolts, then that's going to elicit the opening of voltage-gated sodium channels that are everywhere here, although they're not pictured. Same as in the action potential, and it's going to elicit a new action potential that will propagate all the way down this second cell, down its axon, to the next cell in the chain, or to its target. Let's watch an animation on the synapse.
Action potentials arriving at the presynaptic terminal cause voltage-gated calcium ion channels to open. Calcium ions diffuse into the cell and cause synaptic vesicles to release acetylcholine, a neurotransmitter molecule. Neurotransmitter molecules, acetylcholine, diffuse from the presynaptic terminal across the synaptic cleft and bind to their receptor sites on the ligand-gated sodium channels. This causes the ligand-gated sodium ion channels to open, and sodium diffuses into the cell, making the membrane potential more positive. If the membrane potential reaches threshold level, an action potential will be produced.
Now I want to talk a bit about the specific neurotransmitters that are released into a synapse. First, we're going to start with the excitatory neurotransmitters, and this is the ones which, when they bind to their ligand-gated channel, they cause those channels to open. They cause a new action potential to be triggered in the postsynaptic cell, so they're excitatory. Excitation in this neuron leads to excitation in the next neuron. Glutamate is the main excitatory neurotransmitter of the central nervous system, so the brain and the spinal cord, and it's used in all sorts of different functions. Acetylcholine is one which is involved in muscle signaling. So acetylcholine is the neurotransmitter that is released when a neuron synapses onto a muscle cell. Acetylcholine is going to open up ion channels that elicit contraction of the muscle. Noradrenaline is closely related to adrenaline, but this time it's acting as a neurotransmitter rather than as a hormone in the blood. And last we have dopamine, best known for being the chemical of pleasure, but it's also involved in other functions in the brain as well. Notice that these all have a very similar chemical structure, yet they have different effects and they work in different parts of the brain. There is a very high specificity of these ligand-gated channels. They will only bind with their neurotransmitter and not just something that looks a little bit similar. So these are all excitatory. The brain also has inhibitory neurotransmitters. And that would be GABA, gamma-aminobutyric acid, and glycine, the smallest amino acid, also acts as a neurotransmitter in the brain. Both of these chemicals, they're going to be released from neurons which are inhibitory. This means that when a neuron releases GABA onto its postsynaptic neuron, it is inhibiting activity in the postsynaptic neuron. Same with glycine. It's important that the brain is able to do both excitation and inhibition to form its complex networks. It's like a computer network. There are many connections of different strengths, and some are inhibitory, some are excitatory. I want to tell you about some disorders of these neurotransmitters. Acetylcholine, you'll remember this is the one involved in muscle contraction. There's a toxin called botulinum toxin, which is a chemical produced by a bacteria, and botulinum toxin affects the transmission of motor signals because it inhibits the binding of acetylcholine to its ligand-gated channel. So let's back up a bit here. At the synapse, acetylcholine needs to bind to the ligand-gated channel, or we can also call that the acetylcholine receptor, in order to elicit an action potential in the postsynaptic cell. What botulinum toxin does is it binds to this channel and it prevents acetylcholine from binding and activating the channel. So the channel is going to stay closed and it's not going to open, no positive ions are going to be able to move through, and the cell will not depolarize. There will be no action potential in the postsynaptic cell. When this happens in the motor neurons, it results in a paralysis of the muscles. Botox is the commercial name for botulinum toxin, and it's used cosmetically for wrinkles. A wrinkle is when the muscle in the face becomes involuntarily chronically contracted over time, and injections of small doses of Botox will essentially paralyze those muscles, partially paralyze them, and reduce the appearance of wrinkles. Botox has also been used in cases where somebody has involuntarily contracted muscles, such as people who are cross-eyed. Injections into the ocular muscles will reduce that. And Botox has also been used in cases of bruxism, where you grind your teeth involuntarily at night. Drugs can also affect the inhibitory neurotransmitters. Valium is a popular drug used for anxiety, and Valium is in a family of drugs called the benzodiazepines. These drugs, all in some way or another, they work to amplify the effects of GABA in the central nervous system. GABA is an inhibitory neurotransmitter, so when it's released by a neuron, its job is to inhibit the next neuron in line. So if we're taking drugs that increase the effectiveness of GABA, we're essentially inhibiting the neurons in the central nervous system. Heath Ledger, when he was found dead, he had a lot of benzodiazepines in his system. This caused the suppression of the central nervous system, including the autonomic functions, so the functions that we don't have to think about doing, like breathing and maintaining our heartbeat. Those were suppressed, leading to his death. Michael Jackson as well, when he was found in his home dead, he had just a whole host of different benzodiazepine drugs in his system. The last example I want to show you is this pathway. This really should say a partial pathway of voluntary movement. But so this is a simplified version of how our body elicits movement from our brain. Each of these squares is a group of neurons in the brain, and they all interact with each other. Some in green, they are delivering excitatory signals, so the substantia nigra stimulates the striatum, and these red ones represent inhibitory signals, so the striatum actually inhibits the globus pallidus. This is getting quickly to look like a computer network, and to coordinate these complex efforts and intricate signals, there are multiple pathways through the neurons. But ultimately, the job of this system is to initiate the correct movements in the muscles, and that's going to come from the cortex. It's the cortex, the motor cortex to be specific, that will send the signals to the muscles saying which ones should contract when and at what frequency. In Parkinson's disease, one that's characterized by uncontrolled shaking movements, there is a lack of activity in the substantia nigra. These neurons, either they are dying off or they're not producing enough neurotransmitter, and they use dopamine as their neurotransmitter. One of the earliest treatments for Parkinson's disease was a drug called L-Dopa, which is a precursor to dopamine. So if patients would take it, their bodies would naturally metabolize it into dopamine, and they would have more activity from these neurons, helping their symptoms, helping the brain to coordinate the right signals into the cortex. More modern treatments for Parkinson's disease involve reducing the activity in some of these later higher parts of the brain. Either the thalamus or the globus pallidus can be inhibited in order to prevent the abnormal signals from reaching the cortex. Michael J. Fox, when he was diagnosed with Parkinson's, this was even before he even came out publicly about it, his doctors recommended that he have a thalamotomy, and that is the removal of a small part of the thalamus. And the hope was that by removing part of the thalamus, it would stop some of the irregular signals that the thalamus was sending the cortex. It was those irregular signals that were causing his shaking. So it was successful in treating some of his symptoms at that time, reducing the amount of irregular input to the cortex. More recently, instead of removing part of the brain, because that's considered kind of invasive and not a lot of people want to go for that, we've developed a treatment called deep brain stimulation, and that involves surgically opening up the brain, inserting electrodes which feed into either the globus pallidus or the thalamus. And these electrodes are going to create signals that overpower some of the activity, the natural activity of these parts of the brain. So the idea is to reduce activity in either the thalamus or the globus pallidus so that these parts of the brain aren't sending so much irregular input to the cortex to reduce the shaking movements. So here you can see why the brain has so many multiple pathways and why it's important to have both inhibitory and excitatory neurons. Next session, we're going to zoom out into the entire nervous system as a whole and see how it coordinates its functions. Thanks for watching.